Hi everyone, and welcome to our third DigitAF webinar. So just about DigitAF, this is a European project with the objective of developing tools to boost agroforestry. And actually, this is the last year of the project, so we have several outputs to present, and that's why we choose to offer free webinars to you to share our results, and hoping it will help your agroforestry journey. And before introducing our speakers for today, I would like to let you know a few points. The first one is that this webinar will be recorded, and so it will be available online after the end of the session. And at the end of the webinar, we will also have a dedicated question and answer session. So don't hesitate to leave your questions on the chat or on the Q&A session. We have a Q&A session on the chat, but on the chat is okay too. And we will answer the question at the end of the webinar. And the third point is that, unfortunately, we do not allow AI assistants, so I will delete them. So today, you will be able to know more about our biodiversity and soil health outputs and tools, thanks to our two speakers, which are Jari Vandendriessche, which is a PhD researcher actually at ForNaLab in Gent University, and also we have Michel Den Herder, who is a senior researcher at EFI, which is the European Forest Institute. So thanks, Jari and Michel, for being our speakers for today. And Jari, I think I will leave the floor to you so you can present, and I will stop sharing my screen so you can share yours.
Okay, thank you, Roman. Thank you for the introduction. So, like Roman said, I'm Jari Vandendriessche. I'm a PhD student at Gent University, and my topic lies completely within DigitAF and is based on the biodiversity aspect performed there. So that's also what I'm going to talk about today. Sorry, yes, it works. So I'm going to start with what we actually already knew about biodiversity aspects within agroforestry systems. And yeah, as for first, what is biodiversity? It's a very complex topic. So total biodiversity in an agricultural system is actually consists of introduced biodiversity. So the biodiversity as in crops, as in trees that are introduced for production types or aesthetic function. And then you have associated biodiversity, which is then the spontaneous organisms that are associated with a particular system or with the environment as such. Biodiversity can also be divided in different functional types. And with these functional biodiversity, we mean that certain groups of species have an influence on soil health, for example, which is where Michael is going to talk about, but also pollinating aspects and, for example, natural enemies that do a certain pest control in an agricultural system. What do we know about biodiversity gains through agroforestry implementation? Overall seen, so the figure that I'm presenting here is actually a summary of different synthesis studies that we have performed within DigitAF. And we can conclude that overall it's quite positive, but effects are mixed throughout the system. So there is some difference in design of an agroforestry system, but also among different taxonomic groups, etc. And we also found out that there are you see a lot of blank panels, so there are quite some data gaps on this level of analysis. And what drivers do we suppose that are occurring in agroforestry systems? So as for a first, the type of a system, so within agroforestry, which is highly diverse, you have food forests, silvopastoral systems, silvo-arable systems. There is expected that there is quite some difference in the behavior of biodiversity gains or losses throughout these systems. But also, like I said, throughout different biodiversity groups, like pollinators or birds, and also at what aspect of biodiversity you're looking at. So you can look at the abundance, so the quantity of organisms that occur on a certain field, but also on the richness, so the amount of species that you have on a field. And for the rest, I can relate to the figure to the right. So that's actually a figure that was published by Florian Cléty in 2023. And there is very clear on what design aspects and what environment aspects are important to agroforestry systems. So for example, the implementation of a woody feature and how you're managing it gives quite some results. So for example, the structure and age, the species that you can implement will alter biodiversity outcomes. The same goes up for environment, so the type of climate that's around and the landscape context, even though you even can't influence that, and the kind of management you are applying to the field. Now, within DigitAF, what did we actually measure as a fundamental research? So what kind of case studies did we carry out? As for a first, we focused on Flemish landscapes, so in Belgium. Now we selected across 27 different landscapes across a gradient of really strict landscapes with only agricultural fields and buildings, for example, to landscapes with more semi-natural habitats. And the aim, therefore, was actually to look if there was a difference in if the effects of agroforestry on biodiversity levels, if that was consistent throughout these landscapes, because there are some other research and environmental measures that there is some mitigation effect, so that whenever a landscape is more rich and actually harbors more biodiversity, that effects generated are slimmer. What did we actually do? So we looked at cereal fields with bordering tree rows, and then we sampled in the tree row in a cropland plot and in a control plot, so in a grassy margin. And therefore, we really wanted to maintain a multi-taxonomic scope, so a scope where we could really gather a broad influence on overall biodiversity present on a field. So we applied quite some different techniques to catch as much groups as possible, and really with the aim to get a view of the functional diversity present. The year after, we expanded this approach and we sampled on a European context with agroforestry fields in the UK, in Belgium, in Czechia, southern France, and Portugal. And in each country, we then selected 6 sites within each an agroforestry field and a proper control field, where all these fields were 10 years old or older. Also here we maintained the aim of a landscape gradient, so we really wanted to explore if these agroforestry effects were consistent throughout the whole system of the whole universe of agroforestry systems. And for this study, we also integrated both silvo-arable fields and silvopastoral fields, depending on the location we are sampling. So for example, in Portugal, silvopastoral fields are much more common. And also here we try to maintain the multi-taxonomic scope, but here we even added 2 other metrics to sample even broader than. This is just a list of all taxonomic groups that we wanted to investigate. As you see, there is still a lot of work been done at the moment, but I will show you in the next slides some yearly results we can gather from now. So yeah, what did we learn? For the Flemish set, we've monitored overall quite positive results. So for most groups, we got positive responses for both species richness and abundance. And for a few groups, we found no effect. So also not a negative effect, but just it's the same. Cropland holds the same species richness and abundance as the tree row itself. What happens in terms of landscapes, and that was quite remarkable, is actually that we didn't find any clear effect. So there is either no effect of different of landscape context on the effects. So the gains, in this case, gains gathered by implementation of agroforestry, or it's quite weak and not being captured in the experimental design we applied. Yeah, we only had 27 fields, so that's still a possibility. But even then, if it was really an obvious effect, we would have noticed it by now. So in conclusion, agroforestry effects might be similar throughout to all those landscapes and might be quite stable. For 2024, I can only present the differences between biodiversity on the agroforestry fields and the control fields. And also there we found mostly positive effects. So across a lot of taxonomic groups, only spider abundance was lower in agroforestry fields, but that's actually a drawback from the sampling we applied, because on arable fields, spiders, for example, are much more active and can crawl quite a lot, where they are easily caught. Now, this is the most important part of my presentation now. What about the biodiversity tool? So the work in DigitAF conclude both the experimental frameworks I just showed now, but also the construction of biodiversity tool and the reason why we constructed that biodiversity tool. So that's the second part of this presentation. At first, we had a look at what tools already existed and if they were suited to agroforestry and the biodiversity, especially in a really diverse way. And then our primary conclusions were that actually existing tools were not really agroforestry tailored. So some tools really incorporated the presence of trees or shrubs, but not in an agroforestry implementation, etc. And other studies also only represented an overall biodiversity estimate, while we wanted to go broader. So then we developed a biodiversity model or tool that really could incorporate those things. Now, what target did we aim for? So we really aimed for a field scale assessment so that you actually could start from a baseline field with no agroforestry implementation, and that then could simulate what would happen if you implemented an agroforestry setup on that field. So actually we wanted to really calculate the delta, so the difference in biodiversity levels you could achieve by the implementation of agroforestry, and that both for silvo-arable as for silvo-pastoral or other systems. How did this relate with the collected field data? We really just added the collected field data in the primary step, so the training step of the tool. And also there for the tool, we really want to make a multi-taxonomic approach so that you can really see the difference, how different taxonomic groups react on agroforestry implementation. So that's also, it's something very important. Now, if we refer back to the figure of Florian Cléty, what effects of what implementation design environmental factors for agroforestry implementation did we incorporate? Well, as first we integrated climate. We try to integrate the age of the system, so the age of the trees that are implemented, a landscape context, the width between tree rows in an agroforestry field, and then the structure of a tree row with then the number of species applied and the presence of a shrub layer. What data did we use? So as for first, we indeed used the data from our fieldwork, but also from 60 other case studies that we gathered throughout the research we performed by reviewing meta-analysis, so really meta-synthesis of existing case studies. And this resulted in 62 studies that we implemented over 16 countries, and this included 175 different agroforestry sites. So that's quite a lot. Overall, the effect in this database hinted at a biodiversity gain of 60.7%. So an obvious positive effect of agroforestry, but it's still quite heterogeneous, as you see in the plot. So the plot to the right is all different studies we chose and the effects monitored throughout that case study itself. So you see some case studies do report negative effects, while most, actually, I think 1/3 report negative effects, and then 2/3 goes to the positive ones. So yeah, overall, it's quite positive, the story. Now the tool itself, as for now, it's called Agroforestry Biodiversity Gain Estimator. It's still a beta version. You can see the QR codes and the link to the right, so you can properly retrieve it after this webinar as well. So the first page of the tool is really an introduction page about what we aim for, what the tool does, what background it is, and what that data is integrated. If you proceed, then you can select the agroforestry system you want to simulate. So in this case, I chose agroforestry in broad view, but you can also select silvo arable systems. And in future, the aim is also to integrate silvopastoral systems. And then you can select the different models we have in store for now. So in this case, we have a model for invertebrates in general, and then the abundance and activity, so really the quantity of the animals that are present in a field. We also have a model for above ground invertebrates, so all flying invertebrates like beetles, butterflies, etc. And we also have one for pest species, so really herbivores that live in an agroforestry field. Those are, and not all of them are really harmful to agricultural sites. A lot of them are just grazing on the trees, etc. And therefore we have a species richness model. Now, what does our tool do? You can assign different climates, different landscape contexts, or in this case, it's the percentage of woody habitats within a radius of 1 kilometer, the woody species richness, allele width, etc. And this returns then a table. I will go back. So in this case, I selected the 3 models at once. That's also the reason you have 3 lines in the table. And then each line gives really the prediction of gains or losses that are generated due to the implementation of the system you just assigned. So in this case, everything is positive. And it also gives some indications on the errors been made in the model. Because since biodiversity is still very heterogeneous, as it's agroforestry, these confidence intervals are quite broad. What do we present as well is some plots with then the biodiversity gain, so the percentage gain or loss generated throughout the age of the agroforestry systems. So yeah, it's quite clear that there is a increasing link with age. Now I will loop to really investigate all different layers of the tool. I will just do some scenario testings where we every time I will change just one parameter to see what changes within the model. So in this case, the baseline we chose was a model for we are simulating a Mediterranean climate with a woody habitat fraction of 10%. Just one tree species that's implemented with the distance between 3 rows is 10 meters without trips, and the age is 15 years. And then the result is what you see to the right. So in this, it's quite obvious that age has a positive effect on biodiversity gains. Now we will change climate type to temperate, and then we see a sudden drop in the effects of biodiversity. Still, this is just a model, but it's still remarkable. So apparently, this simulation says that Mediterranean climates have more improvements for biodiversity levels. Secondly, we will change the number of tree rows. So now we change it to 2 species implemented, and you will see a very steep increase again. If we alter the woody habitat fraction, you will you see a small decrease. So apparently landscape has so increasing the woody habitat around simulates a small decrease of biodiversity gains. Increasing alleywidth has almost no effect, but there is a slight decrease as well. If you look very carefully, you see it decreasing a bit. Also important, it's very obvious that abundance and activity models respond more volatile than the richness models. And then the last one is what does happen if we integrate shrub species in our tree row. And then it's quite mixed. So for example, the species richness model, still you see a small decrease, but still responds quite logically. But the abundance activity models almost drowns to minus 100%, which is ecologically actually not interpretable and just remains a question mark. It is something we have to figure out still. Now, what can we consider if we look at these different scenarios? Is yes, we still have, these are still models, so simulations, and which typically happens then is that you have an over and underestimation extrema of simulations, because it still tries to maintain a trend, even though this trend is ecologically not interpretable, of course. And we also see, like I said before, is that the abundance and quantity models respond quite volatile. So that's something very obvious in the really astonishing changes in climate and shrub presence. But we do believe that the model and the simulation give a fair estimate on what directions there are for the biodiversity drivers. So for example, the direction of landscape is the direction of woody species. That's if you integrate more woody species, you expect a more biodiversity gain. We expect that these kind of directions are quite good. So this is the tool that we constructed in DigitAF. Now, of course, we really want to give an overview of what kind of tools you have around for of estimating biodiversity levels on your own agroforestry system. So what other tools are options? as of yet. That depends a bit on what you really aim for. So, for example, they are quite good. So if you really aim to identify what species you have on your farm or your field, you can have a look at, for example, Identifier, which is a photo identification app, which is very good for within a European scope. And if you go to the more extreme regions, I think of very Mediterranean sites, boreal sites, for example. iNaturalist is working on the same way, but has a slightly weaker model. If you want to, for birds, for example, you have Merlin, which really identifies bird focalizations around. If you really want to aim for biodiversity assessment of your system, then FarmTree and Cool Farm Alliance are also quite good options. With the mention that Cool Farm Alliance is not specifically tailored to agroforestry systems, but makes an overall farm assessment, which also integrates biodiversity. To give a small overview of all these different tools. So, for example, this is how Identifier works. You take a picture within the app, and it gives just a result with what the algorithm thinks you're observing. So with good pictures, we can get very good results as well. It also incorporates expert validation, and it feeds into an open in a database of observation.org. So all data is you can find it on the internet. For iNaturalist, it works quite the same. But here you see, so like I said, the algorithm is a bit weaker, so you don't have clear percentages that's assigned to a different of an identification, but it controls for whether the species is seen in the region or not, which is also very good to give a notion on what's around. And also this implements expert validation. And for Merlin, it's Merlin also has a photo identification tool, but since iNaturalist and Identifier work on the same way, I'm not going to show that. Most importantly, here is the bird focalization identification that you have, and then you can really see what birds is singing at the time if you record just on the site. In terms of biodiversity assessment, FarmTree really gives you the option to indicate your plot characteristics. So it's also plot-based. You can also make it a bit broader than that. But then you really just complete all information on your field. And then the results are this is called Shannon diversity. It's actually a metric for biodiversity in global, and then it gives you how biodiversity levels differ throughout the age of the agroforestry system. You can also make comparisons between different scenarios in that tool, so it's quite interesting. I also added the link here. For Cool Farm Alliance, there you have a kind of questionnaire you have to fill in for your farm, which really goes from the products that you make, the practices you do, and then the habitats that you have on your farm. And you just have to integrate really questionnaire based. And the result is some, what percentage of it's quite hard to interpret, but it's actually 100% is the maximum you can do for a specific biodiversity group, and it assesses how effective your implementations are towards different groups. So that's it. Maybe I have one disclaimer yet. So as I'm trying to make clear is that biodiversity is very variable, and that it's really a local biodiversity depends on what species pools are present within a surrounding landscape and the environment to court. So assessment tools are only capable of simulating average trends. So whatever you've seen here is really just the average. One particular site can really respond quite differently. That's also what I've tried to show in the database. So if you remember the different studies that respond quite diversely, that's actually because it's very variable. So all we can try to simulate is the average trend, which is in case of agroforestry quite positive. Thank you.
Thank you, Jari, for your presentation. We have lots of questions, but we will answer them at the end of the webinar. So now we will just go on with the next presentation from Michel.
Yeah, thank you for the introduction. Yeah, that's really nice. I will start my presentation now. I hope you can see it. I think now you should be able to see it. So yeah, I would like to tell you a little bit about the soil health assessments, what we did in the DigitAF project in the Living Lab case studies. So this was a very big effort. So almost everybody from the DigitAF project contributed, especially the Living Labs. So first I give a brief introduction why soil health matters and why it is important. Yeah, so what is a healthy soil? So a healthy soil is a soil that can support crop growth without without degrading the soil and causing other harm to the environment. So a healthy soil, it should be suitable for agricultural production, and then it should also stay stable. And so it should stay stable, so in the same state, or the soil health should be improved, and then you should be able to measure that in a physical, chemical, or biological property over a certain time period. So it shouldn't have so your activity shouldn't have an adverse effect on the ecosystems. So soil health is very important. Currently, about 30 to 62% of the agricultural soils globally face some form of degradation, and that is really threatening agricultural productivity and also biodiversity on a global scale. And also in Europe, this happens actually at quite a large scale. Here on this map, that's from the EU soil monitoring dashboard. So also in Europe, there's quite a lot of degraded farmland where there is some problem with soil health. So soil degradation processes, it can be like soil erosion, loss of soil organic carbon, nutrient losses, decline in soil biodiversity, salinization, soil compaction is a big problem, contamination, and soil sealing. And land conversion has a very big impact on soil health. So that is if you change one land use to another land use. For example, if you change forest to agricultural soil or pasture to arable land, and also the type of management that you carry out on your land, that has a very big impact on the soil. Agroforestry has been proposed as a promising method for improving soil health. So previous research has shown that it can benefit soil structure and also increase soil organic carbon. Yeah, to test that. So in our soil health assessment in DigitAF, yeah, we wanted to evaluate different soil health indicators in our living labs. So we have 6 living labs and 2 participating countries. Living lab in Finland, UK, Netherlands, Germany, Czech Republic, Italy. So those are the living labs, and other countries which joined. They have a kind of living lab, but they're not an official living lab. But they also, yeah, they also helped in the soil health assessment. So as in Spain and in Portugal, we also did the soil health assessment. So yeah, we covered a large part of Europe in very different environmental conditions. And the aim is also that we come with a uniform protocol of doing soil health assessment and yeah, sharing the data openly for that it can be compared for future studies. Yeah, because yeah, these sites they span quite a big part of Europe, and the agroforestry types they were also very different. So we started with yeah with developing a soil health assessment protocol. So my colleagues they were yeah very active in that. And yeah, because we have so different agroforestry systems in our living labs, yeah, the aim of this soil health assessment protocol was to give some guidance to the living labs how to carry out the soil health assessment, that it's done in a similar way, and that it's that it's a big literature review, reviewing how earlier assessments have been done, so that it's yeah, with the aim of harmonizing these assessments. So in an agroforestry system, you can have like the trees can be arranged in rows, like in an alley cropping system. So then you do your sampling. Then you're sampling the 3 rows, but it can also you can also do area-based sampling. For example, in silvopastoral systems, there we compare the control and a treatment area. So an area with agroforestry compared with an area without agroforestry. And then for the carbon and the pH and the nitrogen, there we took composite samples. So then we took one sample in the middle, and then in all directions we took another sample. And then the samples were analyzed separately, so then you can get a better estimate in each point. Yeah, so here you see the different types of agroforestry systems from the air. Yeah, so in the different countries, there were like, yeah, we had silvopastoral systems, and also then row-based systems, so they were analyzed in a different way. Yeah, then based on previous assessment, we selected some indicators what looks yeah useful for assessing soil health. So yeah, from Lucas and the land degradation surveillance framework, these indicators came up. So we chose the soil organic carbon, bulk density, pH, and earthworm abundance. Yeah, so in soil organic carbon, that reflects the carbon sequestration and soil fertility. Bulk density, that gives an estimate of soil compaction, what also has a very big impact on water infiltration and yeah, how crops and trees can grow. pH is important for the nutrient availability and the microbial activity, and earthworm abundance is an indicator of soil health and also activity of soil health of activity of soil organisms. I tell you a little bit about the case studies what we did. So in the case study in Czechia, you see it here. That's an agroforestry system with alley cropping with fruit trees. It's an organic system. Yeah, here you see the results of the soil organic carbon content in different transects at different distances from the tree row. So the most on the left, that is inside the tree row, and then they took samples at 1 meter, 3 meter, and 9 meters from the tree row, and also at different depths, and also in a control area. So you see that in the tree row, the soil organic carbon was the highest, and the further away you go from the tree row, the soil organic carbon slowly starts to decrease. And also with depth, so you see in the top layer, the soil organic carbon content is the highest. And if the soil depth increases, so at deeper depths, soil organic carbon, it gets less. So that's interesting. In Finland, the site in Finland, that was a silvopastoral system. There was a wood pasture with forest grazing. So that's a traditional system. Actually, on this picture, you see what the place looked like 130 years ago. So that time in Finland, there were actually in the countryside much more people, and there was much less forest, and there was more agriculture, and also quite a lot of local industry. And nowadays, many people have left the countryside, so you see a clear difference that the area is nowadays much more forested. So that's quite interesting to see. So in the Finnish side, there we compared grazed forest and ungrazed forest, and then open pastures without trees, so permanent grass with no tree cover. And here you see that the soil organic carbon was the highest in the grazed forest in the top layer, so the layer from 0 to 20 centimeters depth. So the lower depth had less soil organic carbon. The forest also had quite a high level of soil organic carbon, and the permanent grass had much less soil organic carbon. And there was not much difference if it was grazed by sheep or by cows. The earthworms, there were still some earthworms in the grazed forest and in the wood pasture, but actually in the permanent grass, the earthworm abundance was much higher. If we go to Germany, there's a silvo-arable system with short rotation coppice and a silvopastoral system with willow and alder. In Italy, there we have a silvo-arable pastoral system with poplar, and there they took samples in a control area. Then in the area with poplar trees, and then also inside the tree rows. In the Netherlands, there they had also an alley cropping system with dairy production. That was with chestnut and walnut. And in Portugal, they had a Dehesa area, and there they compared forest with the Dehesa area, the silvopasture area, and then open grassland or shrubland. And in Spain, they also had the previous one was a Montado area, and in Spain there is a Dehesa area. But yeah, it's a cork oak system. And in Portugal, it was area-based sampling that they took transects. And in Spain, they took the measurements under the tree and then a bit further away from the tree in the pasture. And in the United Kingdom, there they took also samples in the open pasture area. Then they took samples in the silvopastoral area, and then they took soil samples in the forest area. Now, if we look at the results, so here you see the results from all the countries at different soil depths and in different agricultural practices. So here in green, there you see where there was a difference. So you see that in most countries in the area with the agroforestry trees, the soil organic carbon was it was a bit higher, or in some cases actually quite a lot higher. So that is that we could observe in most countries. Only in Portugal, the grass control, the soil organic carbon was a bit higher, but there was not so much difference between the open pasture and the wood pasture. But overall, there was quite a positive influence of permanent vegetation like trees and shrubs, and that resulted in higher soil organic carbon content compared to open fields. We also see that in Finland the forest area or the silvopasture area had a very high carbon content. First, I thought that how is it possible? It's very high. But then, after doing some research, we thought that there's a very big range in soil organic carbon. So this study is based on the Lucas data set, where they also study soil. And they're so in our study, soil organic carbon ranged from 1.4% in Portugal to 13.6% in the forest control plot in Finland. So in the wood pasture, it was 11% soil organic carbon, and in the forest plot, it was 13% soil organic carbon. And this map, you see that in Northern Europe, the soil organic carbon content is very high. So soil organic carbon is really dependent on temperature and drought. Also, pH has a very big impact, and also the type of vegetation has a very big impact. The pH in the sites in Southern Europe, they were during the measurements quite dry, and the Finnish side, so in the north, the pH was extremely low. And if you have a very low pH, and it's also a very cold place, so decomposition goes much slower. And because of this low pH, it even goes slower. So there's also less activity of soil organisms. So then you get that kind of typical pot soil what you find in Northern Europe. So they're very acid, but also have very high soil organic carbon content. Yeah, if we look at the acidity, so for that so the acidity ranged from 3.8 in Finland to 8.2, 8.4 in Italy. So 3.8 is, yeah, that's extremely low, but that's, yeah, that's probably then caused by this low temperature and an accumulation of biomass, and also the presence of coniferous trees. So that causes low acidity, and that can also, yeah, then the organic material decomposes much slowly, so that's why we see these high amounts of soil organic carbon. But yeah, agroforestry didn't have a big effect on pH. And if we look at the earthworms, agroforestry also didn't have a very big impact. So most sites, they had some earthworms, except in the UK and Portugal, the number of earthworms was very low, probably because it was very dry during the sampling season. So they did the sampling in spring, and yeah, then they tried again in autumn, because then it started raining, but still there were not so many earthworms. And in Italy, Spain, and Finland, there were some earthworms, but not very many. But the Netherlands had a very large amount of earthworms. So that's that has a very good climate for earthworms. Earthworms, they don't like acid soils, and they also don't prefer very dry soils. Then we also tested one tool. So we didn't build a new tool, but we we were looking what kind of soil health tools do exist. And then we came with this tool. It is mostly focused on the UK. So this is a tool by the UK Centre of Ecology and Hydrology. So they made a soil health tool, and people can, everybody can use it, and you can enter there your soil health data. So the same data we took from our sample field. So you have data on soil organic matter, earthworm abundance, pH, and bulk density, and then you see how your field or farm compares to other farms in the same sector. So that's a quite useful tool. So it doesn't give you an estimate how healthy is your soil, but it gives you an estimate how the soil on your farm compares with other farms in the same sector. So then you can think about ways how to improve your management. So here I put here the values of the Finnish farm compared with the average farms, and you see that actually everything was in the soil organic matter was within the range what you expect, and also bulk density was within the expected range, but the number of earthworms was much lower, and the pH was much lower. So we found this quite a useful tool. Then some conclusions. So there were not so many soil health assessment tools. And actually there's still a lot to improve in this tool development. So these tools, they miss the age of the system, certain species, also spatial variabilities, not very much taken into account, and especially agroforestry is not taken into account very much. Yeah, so it seems that there doesn't exist yet a rapid, cheap, and accurate tool for soil carbon and soil organic carbon assessment. So there's still a bit of work to do. Yeah, so it yeah, for example, for carbon certification, what they are planning, it would be very important that a standardized tool is developed, and so that can yeah, that can be yeah, that can help if we want to establish this kind of carbon market. And that would also help for farmers to yeah, to adopt the practice of agroforestry. Yes, so this report what I now presented and the data and the soil health assessment protocol, they are or they will be available on Zenodo and in the DigitAF agroforestry data catalog. So all these DigitAF tools, you can find them on the DigitAF virtual space. So Zenodo is for reports and for data. In the data catalog, there you can find all kind of useful data related to agroforestry. So what I just presented will be uploaded there. But then we have also other tools like on the DigitAF virtual space, like the tool catalog, the LPIS sustainability compass. So there you can see it already works for 9 countries which have a land parcel identification system. So then you can go to your own farm online and then select your field and see. Yeah, you can see different. You can see different things like tree cover on your farm and soil type and other interesting environmental indicators. Yes, on the DigitAF GitHub community, that's more for tool developers. There you can contribute to tool development or suggest updates. And then there's also a photo gallery and an agroforestry map of Europe. Okay, yes, so what can we learn from this? So it looks like that agroforestry really enhances soil health under the trees and near the trees, and it has a benefit for soil carbon and also biological activity, and especially in the older agroforestry systems we noticed a beneficial impact for like the sites in Finland and in Spain. They were the older sites, and there we could see a bigger impact on the soil organic carbon. But yeah, there was a big influence on climate and also soil type. And the indicators what we tested, so the soil organic carbon, bulk density, pH, and earthworms, they were suitable indicators, but yeah, they don't tell everything about your soil health yet. So you still have to, yeah, you have to get a bigger picture. Also, really think about, yeah, the site and the climate, and you have to. So soil health is a really, it's like, yeah, like biodiversity is a very complicated map. It's very complex. Yeah, so you have to take many different indicators into account. Yeah, that was it. Thank you for your attention. Yeah, if there are any questions, then we can discuss about them now, I think. I end the slideshow.
Thank you very much, Michel and Jari. Yes, we have questions actually. If you go on the Q&A part of this webinar, this is a dedicated session. This is not the chat, but another part of the webinar. Okay, so we have one question. I don't know if you can see the question right now, the questions, but I will just say the question and read the questions if you want. So Jari, I think it's for you. We have a question from Lina who said, why was there no data for pollinators in the previous, I guess it was slides in your presentations?
I think Lina is referring to the first slide I showed. That slide integrated a summary of studies that summarized case studies that were performed. At the time that those meta studies, so these studies really integrated the information of different experiments gathered out and performed. None of these studies really integrated pollinators because data at that time was too limited. So there was simply no data on that level for pollinators available. As with the current data of case studies, I think I could present a similar graph for pollinators as well. But still, yeah, if there are more than 10, 15 studies addressing pollinators in temperate agroforestry systems, it will be much. Does that respond to your question?
Oh, sorry. Lina, I've also raised your mic if you want to react.
No, just it's very clear. Thank you. It's just that we don't have that much data so far.
Perfect. And you had another question or so. You asked, Jari, could you please do a simulation with agroforestry for agroforestry system type and pest species in the biodiversity matrix? You said you struggled to interpret the graph generated. I don't know if you can do this right now.
I think I can do it. I will try to share my screen because
yes,
otherwise
I
have to be here. Well, I will just run the app. Hope it works. You need to give it some time. So for agroforestry and only the pest species plot. Like temperate. You mean like this, Lina?
Yes, exactly.
So how do we interpret it? Yeah, I also noticed I will immediately address several questions at once because there are, I think, 3 questions that are explainable by this one. As for first, there was a question, why doesn't the graph start at 0? We actually chose to let the graph start at age 1 to avoid that problem because it's typically is the relation from the just before the implementation of agroforestry to the first year is not linear and it's not easy to model, and that's why we chose to only present the curves from age 1 on, and that's also why it doesn't start at the point of 0 by 0. Secondly, how do we interpret this? First of all, it's species richness that is simulated here. So I saw another question that related to whether it's bad if it's that there is a positive effect on pest species in this case, but it's still species richness. So it means you have more species of herbivores here, but that's quite a logical effect because you are integrating trees and shrubs which are initially not in the field, but each of these shrubs or trees has his own herbivores. So species that really predate on the leaves or even the stem of a certain tree species. And that's actually the effect that you see here. So the positive effect is really the bonus of herbivores attached to the trees you are integrating. Is it bad to an agricultural system? That depends. Yeah, the aim is still to have production of these integrated trees or shrubs. So there will be a slight effect, but I don't think it's really avoidable that you have those new species coming in. Something else that we cannot show here, but what we found out is, now I'm talking about species richness, but actually the driver of pests in an agricultural system is really the abundance of these pests. Other results, which I didn't show now because it's too detailed to do it here, have shown that the abundance, so really the quantity of pests present on an agroforestry field, is lower than it is, and then the quantity is on a monoculture field. So actually the effect behind it is quite positive. Now to interpret this curve, so you see at first the first 10 years the effect of so you have slightly lesser species which assimilated in comparison with a monoculture control, but from I think it's year 15 on, you will have an increase in species richness for these pest species. Does that answer your question a bit? Or do you want more information about how to interpret the plot?
On my side, it's okay. It's difficult for me to maybe understand because it's still a model. So
yeah, it's still a model, but what do we? So yeah, the shaded area is actually the error margin of the plot. So I think it's whether it's loss of species or a gain is actually quite unclear. Just the average says it's a gain of species richness from year 15 on. Yeah.
okay, thank you.
Okay, thank you. So now we have a question from Marie, which is actually the coordinator of the project. And Marie, also your mic is authorized if you want to participate. It will be with great pleasure. So your question was, the tool predicts increase in pest species. Does it mean that natural pest regulation does not work?
Well, that's the thing that I wanted to address just before. So it predicts increase in pest species richness. So that's richness that's really related to the integration of trees, which have their own herbivores. So that's quite expected that you have an increase of species richness, but that doesn't. It's not the same story for abundance. So we see a small decrease with abundance, which is not shown in the tool as of yet. But yeah, we've done these analyses, and that's not the case then. So natural pest regulation does work, I would say.
Okay, thank you very much. Another question for you from Roni. The question is, do you have any idea whether pesticides used on an agricultural field next to a food forest has a big impact on the food forests? Talking about fungicides on the mycelium network, insecticides on the pollinators, and other useful insect herbicides on plant life. What would be good buffer plants haste trees?
That's a very difficult one because it's not really my expertise, but I'll have a try. First of all, it will really depend on the context of the field. So for example, if you have really a water flow from the agricultural field to a food forest, you will have some pesticides that are going along with the water that drowns to the food forest. And then it would be better to have a kind of a buffer strip. The more complex, the better it holds up insecticides, etc. Yeah, but still, it's quite variable. For example, you mentioned useful insects. All useful insects that seek shelter or hibernate in the agricultural field will be affected by the pesticides, and these will also transfer to the food forest. There is not really something you can do with it. But yeah, if there is really a close connection, just make sure that the connection is as dense as possible. And then I mean really a structural layer with small trees, very dense shrubs, and a very dense underlayer to really prevent flowing in. But yeah, more I cannot say about it.
Okay, thank you.
Thank you. Then next question. If agroforestry systems increase biodiversity by an average of 16.7%, which specific agroforestry practices or tree species are associated with the greatest increases in biodiversity? Mathis, also I've authorized your mic if you want to complete or just react to Jari's answer.
Very interesting question. What we have monitored is actually that silvopastoral systems have a bigger increase, but then I have to mention that silvopastoral systems surveyed as of yet are generally older. So we saw a quite positive effect of age as well. Could be that silvopastoral systems are just performing better right now because they are generally older than silvo-arable systems are. But for now, the response will be silvopastoral systems. And then in terms of tree species associated, that's kind of analysis that I did not perform as of yet, because a lot of systems really implement multiple species, and then it's really difficult to disentangle which species has most effect. But yeah, implementing more species generally has a bigger effect, of course. Is that a good answer to your question?
Well, I guess we could just go. Okay, good. I thought I've authorized the mic. Sorry for that. That's great. If that answers your question. Okay, next question. This one is okay. This one too. Question is there by woody species richness, does that mean the diversity of agroforestry trees and shrubs?
Yes, indeed. So that's a combination of both shrubs and trees implemented. Yeah.
Good. Thank you very much. It was a quick question. Other question from Honi. The question is, the accumulators in plant guilds mine minerals and trace elements from deeper layers. Do you have data about soil health from a comparison between an agroforestry field where those are implemented and agroforestry field where those are not used?
That question is for me, yeah, I guess.
It's for Michelle. Yes, actually, this is for you.
Okay, yeah. Thank you for the question. That is a very interesting question. Yeah, we didn't look at by accumulating plants, bioaccumulators, but that would be really interesting. For example, looking at nitrogen fixing plants and their effect on soil health. Yes, so we didn't do that, but we did look at bulk density, and I think I didn't present too much details on that. But in most of the agroforestry sites, the bulk density was a bit lower. So the soil is less compact. So that also means that water infiltration is better and the roots can go deeper. So probably the plants can also more easily get their nutrients from deeper layers. So that can have a very big impact. But we didn't do that. But
Okay.
it's a very nice topic for future research. Yeah.
All right. Thank you.
Great. Thank you.
Thank you for the question. Lina, you had another question. Are you planning to extend these tools to tropical areas?
That's also for me, I guess.
Yes, I guess
Or for
so,
both
because
of us, maybe. Yeah.
maybe.
For both, yes.
Yeah. No, I'm not planning to extend them to tropical areas, but of course that would be nice. But there is a lot of development in soil health assessment, also in tropical countries. So that is that's an area for development. And now we did real sampling, so we really did field work. But there are people are developing methods and digital apps, for example, that you can use your mobile phone for assessing soil health. So that's an area what is really under development still. And thank you for the question. I hope it answers it.
And to add from my perspective, so I'm also not planning to extend it to tropical areas, but the meaning of the tool that we created is really to make it very open source, so that the code underneath and the data set underneath is really just in the open, so that everyone can access it. And then it's possible for other people to just replicate my work with then new studies for tropical areas to add on these. But for us, at this moment, there is not really the idea to expand it ourselves. But it would be nice to indeed do it later on.
Thank you.
Thank you very much for the answer. We have one last question. Do you think DNA soil testing is necessary for agroforestry systems?
Absolutely. This was actually done in some of the sites. It was done in Spain and in Portugal, and I think also in Germany. The German site was very young, so there wasn't a big effect. In the Portuguese site, it was done. That was a simple test. There also wasn't a very big effect because probably it was very dry there during the fieldwork season last year. But in the Spanish side, there was a small, there was an effect on soil microbial activity, and there was clearly a positive effect from the agroforestry system. And in all the other case studies, we used worm abundance, earthworm abundance as an indicator, but that's actually not a very good indicator. So in Central Europe, where you have quite a temperate climate, what is quite wet, there it could be a good indicator, but worm abundance in Southern Europe and in Northern Europe, that's not a very good indicator because in the south it's probably too dry, so earthworms don't enjoy that. That's not their preferred habitat. There were some earthworms, and yeah, in the north it's really too acid. So yeah, there the worm abundance is also not a very good indicator. So it would be super interesting to use to do yeah, assessments studying the other soil microbial activity and DNA assessments. We wanted to do that, but it was not possible because the samples need to be frozen and they have to be transported frozen to the laboratory. So that was too big distance. But it would be really nice to do that. But, yeah, thank you for the question. Yeah, and also, yeah, it would be super interesting because now, for example, in the north, there is this extreme acidity, what microorganisms probably don't like, but there could be other microorganisms what can thrive in that kind of acid environment. So that would also be interesting to see.
Thank you very much. We have one last question from Ishara, who was off during some time during your presentation, Michel. Have you presented QBS, the best soil earth indicators between the tree lines and the middle of tree spaces?
I don't see that question. Can you say it again?
Ishara asked if you presented QBS, the best soil earth indicators between the tree lines and the middle of the tree spaces during your presentation.
QBS.
You can find it in the chat.
Yeah. Chat and Q&A session. I will authorize the mic for Ishara too. In a few seconds.
Ah.
Yeah, I've authorized the mic.
Yes, presented QBS, the best soil and health indicators between tree lines and the middle of the tree species. Okay. QBS. No. Don't know what's QBS. Can anybody help me out?
I don't know the acronym, but it's as a biodiversity soil index. I think it is originated in Italy, but that's all.
Okay. Yes.
You know it?
No. But we did. But this already helps because we didn't use a biodiversity indicator.
I know I looked into it a few years ago, and it implemented different soil organisms like columboles. Yeah, it's a long time ago. But
okay. Yeah.
It gives an index based on what specimens are prevalent in the soils that you survey. But yeah, I couldn't answer the.
Yeah, that would be super interesting. Yeah. Yeah, once at a winter course in ecology, we did some sampling in the snow and in the soil, and there we studied different soil organisms like columbola and also nematodes. So that would be really interesting. Yeah, but yeah, in this soil sampling, we only took the the earthworms and the DNA samples, because we wanted to test a quick assessment method, what is easy and what is comparable with the soil health tool what we wanted to test. But this could be super interesting as well. Yeah, but we didn't do it.
Okay, great. This was our last question for today. If you have other questions, please don't hesitate to write them down on the chat right now. Oh, is someone trying to talk right now?
It's me, Chalal.
Yeah, please go. Go ahead.
I'm asking again question for Mikhail. So what health indicator, soil health indicator parameters were included in the case study you have presented between tree lines and between tree gaps?
What indicators did we study? Yeah, so we studied soil organic carbon, soil organic matter, then we studied the pH, soil bulk density, earthworm abundance, and then some case studies also studied the soil microbial activity. And then, yeah, also soil nitrogen, we took that was also included in our samples. And then there was also taken, in some case studies, the soil infrared spectrum, but that was not done in each case study.
Okay, like related to soil biodiversity. What was the outcome or the conclusion you got from the result?
Well, only in the Spanish case there was a clear beneficial effect of agroforestry. So their biological activity was improved by the agroforestry system. But the other studies, there was no clear effect, probably because it was too dry or the site was too young. So probably in older sites, you get a more pronounced effect on biodiversity. So you have to wait a bit longer.
How old was the span, Phil?
The site in Portugal was not very old. Was it like 20 years old? But the site in Spain, that was more than 100 years old. So that's a very old dehesa.
Okay.
Yeah. And the site in Germany was also quite young. Was it about 10 years or a bit longer?
Okay.
Yeah.
Thank you very much.
So in the long run, then probably have a bigger impact.
I hope so.
Yeah, I hope so too. Yes. Yeah.
Thank you.
Yeah, thank you for the question.
Thank you for all the questions and for your presentation and answers to Jari and Michel. I think it's the end of the session of the webinar for today. The webinar has been recorded, so you will be available to replay it online on YouTube, and we will send the link on LinkedIn and on our website. Yes, I guess that's all for today. Thank you very much, Jari and Michel, for presenting all your stuff on your tools and your results and outputs. Very, very interesting. I hope you like this webinar, everyone, and I will just wish you a great day.
Thank you too. It was a pleasure to be here.
Yes, thank you.